Hello friends, welcome back to our YouTube channel Chemistry Lab. In previous video, we was discussed about hyphenated techniques of mass spectroscopy. Now in this video, we are going to see hyphenation technique of gas chromatography with infrared spectroscopy. So let's start the topic. First, we are going to see what is the principle of this technique GCIR. So let's start. GCIR is a coupling technique in which GC does separation and IR does identification. Here we know the gas chromatography is do separation of a molecules, and IR spectroscopy, or we can say infrared spectroscopy, does find out the what kind of functional groups are present in a sample, or we can say identification of a compound. It identify the compound on the basis of their functional groups, what kind of functional groups are present in a sample. So here two techniques get coupled with an interface to form a hyphenated technique of GCIR. The mobile phase commonly used in gas chromatography, helium, nitrogen, and hydrogen. This carrier gas should be transparent in a IR region. Then the carrier gas should be pure one in order to increase sensitivity. Uh, Otherwise, it interfere in IR spectra. Here, the gas should be pure without any other mixture of gas, and it used in both technique GCIR, because when GC is connected to IR, the carrier gas carry out the sample with each other, and that carry sample get passed through IR spectra for identification of a molecule. So here the gases should be pure one, not mixture of gases, and samples should be IR active and non-decomposable in mid-IR region. Here the sample we use that is IR active and non-decomposable for this technique. Speed of carrier gas and sample should be adjustable for clear absorption of IR. Here we use two hyphenation technique or interface. Here two interface are used. One is flow cell method and second is cold trap method. So first we discuss about what is the working of this technique. After that we are going to see interfaces. So the instrumental diagrams is here. Here the sample get injected in GC column, or we can say injection port of GC. Here the sample get enter in GC column, and carrier get with combined with and interact with the sample, and sample get passed through the IR detector for detection, because with the help of interface, uh, light pipe interface or flow cell interface is used in this technique. And so the about procedures, how procedure is work for GCIR, and how the GCIR technique is work in this technique. So here the sample get injected and sample get in GC column. After GC column it get to interface to it go to detector, and detector should be detect the sample, what kind of functional groups are present. The effluent from GC column is directed into light pipe cell. This light pipe is an interface, and This interface is a very important. GCIR consists of spectrometer which contain an interferometer which modulate IR beam source. The modulated IR beam is focused on a heated light pipe cell through which separate GC compound transfer. So an interferogram is generated when IR signal receive a detector and this signal is sent to computer for further processing. Since IR spectrometer is non-destructive, the GC influence can also be directed from a light pipe outlet to conventional GC detection system like flame ionization detector. This permits spontaneous detection of normal chromatogram of corresponding IR spectra for each compounds. Now we are going to see second now we are going to see the interface, what kind of interface use. There are two types of interface use, gas light pipe interface and cold trap interface. So first we discuss light pipe interface. So the light pipe, this is the diagram of light pipe interface. The sample gate passes through the gas chroma gas chromatography column inside the interface. This is the interface. The interface is made by the pyrex tube and this pyrex tube to sample gate to the IR detector. This is the IR detector and this is the GC column to sample gate travel. So let's start with the topic interface. The interface between GC column and IR detector is a narrow light pipe having length 10 to 14 centimeter. Is the length of light pipe interface is 10 to 14 centimeter and inside diameter is 1 to 3 millimeter. The diameter of light pipe interface. The light pipe consists of pyrex tube and this pyrex tube is internally coated with the gold, pure gold. The light pipe is heated in order to avoid condensation of sample molecules. Light pipes are designed to maximize the path length, enhance sensitivity, while minimizing the volume of laser band bonding. Gas spectra contain fine structure which is absorbed in a light or solid spectra is the absence of band due to the intermolecular attraction. Cold trap, this is the second part or we can say second interface in GCIR, cold trap interface. The interface is based on the trapping of analyte prior to the analysis. The gas chromatographic effluent is continuously deposited through a heated small diameter capillary on zinc plate coated by the liquid nitrate. The zinc plate is moved to transport the condensed sample. An IR instrument, the IR detectors are directly sensitive to liquid nitrate. This diagram is represent the whole interface of cold trap system. or we can say cold trap interface. This is a similar to light pipe interface, and both the techniques are same, only small difference are there. And why we use this technique, GCIR? GCIR is a very important technique for the identification of monosaccharides and amino acids, fuels, polymers, and natural fragrance, perfumes, and other materials. Drug analysis, food samples, blood samples can be detected with the help of GCIR technique. Coupled GCIR techniques are very important. Nowadays, all the industry use this hyphenation techniques for better result and less time consuming these techniques. So these two questions are there. What is the role of light pipe in this method? I explain the neat level diagram how IR can be coupled with GC. Thank you for watching this video. If you like this video, please like,